Okay, and now let's go through the lecture. So the first lecture is an introduction to the endocrine system physiology. Because it is introduction, let's start with the definition of the hormone. Hormone is a chemical substance which is produced by cells, tissue, and organs that controls and regulates function of cells, of the target cells. This includes metabolism, growth, development, proliferation, electrolyte balance, reproduction, behavior. So we may say that hormones regulate functions of all cells within our body. Based on the structure, sorry, based on the structure, the hormones can be classified into three different groups. The first group of hormones are derivatives of amino acid tyrosine. The next group are proteins and polypeptides. And finally, the third group, steroids. Let's start with the first group. Derivatives of amino acid tyrosine are synthesized by two glands, by thyroid glands and adrenal medulla. Remember, please remember, adrenal medulla, but not adrenal cortex. So derivatives of amino acid tyrosine are produced once again by thyroid and adrenal medulla. Thyroid produce T3 and T4, so triiodothyronine and thyroxine, T4. Adrenal medulla, as you all know, produce epinephrine and norepinephrine. The second group of hormones are proteins and polypeptides. As you can see on this graph right now, almost all hormones that are produced in our body are classified as proteins or polypeptides. So it depends on their size, obviously. So among this in this group of hormones, we can distinguish hypophysiotropic hormones, which we are going to talk about a lot today, oxytocin and antidiuretic hormone, well known by you from the kidney physiology lecture. Then we have hormones which are produced by and released by the anterior pituitary gland, like tropic hormones, including growth hormone and prolactin, pancreatic hormones, insulin, glucagon, parathyroid gland hormone, which is parathyroid hormone, which is responsible for calcium metabolism. Next, the other hormone which regulates calcium serum concentration, which is produced by thyroid, calcitonin. Some of the hormones that are produced by placenta, like human chorionic gonadotropin and human somatomammotropin. There are also other hormones that are produced by placenta, but they are not peptides or polypeptides. They are steroid hormones. As you know, heart also produces hormones, including erythropoietin and atrial natriuretic peptide. Stomach produces gastrin, which together with secretin and cholecystokinin are very important for the proper functioning of gastrointestinal system tract. And finally, leptin, which is also crucial for GI tract, is produced and released by adipocytes. When we look at not only into the structure, but also we consider the chemical properties of the hormones, we get to know it becomes obvious that amino acid derivatives, proteins, and polypeptides, they all have similar chemical properties. There is one exception to this rule. Although T3 and T4 are the derivatives of tyrosine, they act similar to steroids, which we are going to discuss in a moment. So all derivatives of amino acids besides T3, T4, and all proteins and polypeptides have similar chemical properties. So first of all, they are water soluble. They are hydrophilic, which means that they cannot go through, they cannot pass any biological membrane, including cell membrane. That's why the receptor for derivatives of amino acids, proteins, and polypeptides are located on the surface of cell membrane. So they are called membrane-bound receptors. The hydrophilic hormones, so the water-soluble hormones, can be stored within the endocrine cells. The ability of the endocrine to store the hydrophilic hormones plus the mechanisms of their action, including membrane-bound receptors, results in very short onset of hormone action. So all derivatives of amino acid besides T3, T4, all peptides and polypeptides are characterized. They induce an effect which has a very short onset. As you will find out in a moment, most of the membrane-bound receptors act via directly or indirectly. They act via protein kinases. So the effect is due to phosphorylation of different proteins, and because of that, the effect of hormone action is short-lasting. So there is a quick onset of effect, but the effect is short-lasting. And finally, all hydrophilic hormones can be, with three exceptions, actually, hydrophilic hormones can be transported by blood freely, but we'll get back to it in a moment. Steroids are a third and totally different group of hormones. Steroids are all produced from cholesterol, so it doesn't matter what kind of steroid we are talking about. All steroids are produced from cholesterol. They are produced from adrenal cortex, ovaries, test, and placenta. So, adrenal cortex produces glucocorticosteroids like cortisol and mineralocorticosteroids, mainly aldosterone. Okay, ovaries produce or maybe the follicles within the ovaries, they produce and release estrogens. Remember that there is no estrogen, there are estrogens, a whole group of hormones, including estradiol, estriol, and progesterone. Tests produce testosterone, and placenta produces estrogen and a huge amount of progesterone. As I said earlier, all steroids are produced from cholesterol. So because of that, they have hydrophobic. They are all hydrophobic. So they are lipid soluble, and because of that, they can freely pass through any kind of biological membrane, including cell membrane or nuclear membrane. Uh, because of their hydrophobic feature or character, the receptor for steroids or the receptor for hydrophobic hormones is located within the cytosol or within the nucleus. And in general, we can say that all hydrophobic hormones are responsible for the regulation of protein synthesis within the target cell. As you know, the protein synthesis requires transcription, translation, post-translation modification, which is all time-consuming process. That's why the onset of steroid hormones effect within the target cells is late, is prolonged, and but the effect, because of the new proteins that are present within the target cell, is long lasting. So prolonged onset of effect, but the effect is long lasting. The prolonged onset of hydrophobic hormones action is not only due to their mechanism of action and the nuclear receptors, but also is due to fact that that steroid hormones cannot be stored within the endocrine cells. So we may say that hydrophobic hormones, steroid hormones, are synthesized de novo, so according to the demand. Okay, and finally, hydrophobic hormones, because they are not water soluble, cannot be transported by blood freely. Besides the classification based on hormone structure, chemical properties, hormones can be also classified according to their mechanism and site of the action. There are four different mechanism and site of hormones action. So first is endocrine mechanism. In case of endocrine mechanism, the endocrine cell and target cell are far away from each other, so they are spatial separated. The endocrine cell produces hormones, which are then released by exocytosis to the blood vessels. The blood vessels allows to transport the hormones with the blood to the target cells, and then the receptor binds to the hormone binds to the receptor within the target cell, inducing the effect. I think that almost all hormones act via endocrine mechanism. So you know about renin, about antidiuretic hormone, which is produced and which is produced in hypothalamus, released from posterior pituitary gland, and then acts on kidneys. So you know that this is actually an endocrine action because antidiuretic hormone requires blood transport from posterior pituitary gland to the kidney. What else? Insulin, glucagon, T3, T4, basically all hormones can act endocrine. In the second mechanism of action, we also have two cells. So there is an endocrine cell and target cells. However, in case of paracrine action, the target cells is a neighboring cell to endocrine cell. So endocrine cell and target cells are neighboring cells. Okay, so again, the endocrine cell produces hormones that are that are secreted via exocytosis to the extracellular fluid. The hormone which is released to the extracellular fluid then binds to the specific receptor within the target cell, inducing the effect. So there are several examples of the paracrine action. So the most well-known example of paracrine action is the effect of insulin on glucagon production and release, and the other way round. Okay. So this is the paracrine mechanism. In the next two mechanisms, the endocrine cell is actually the target cell. Let's start with the intracrine action. Sorry for that. Intracrine action. So during the intracrine action, the endocrine is a target cell. Endocrine cell is a target cell. The endocrine cell produces hormones, which are almost mostly released from the cell to the extracellular fluid and may act paracrine or endocrine. Okay, a small portion of a hormone remains within the endocrine cell and then binds to the specific receptor within the endocrine cell. Hormone binding to the receptor within the endocrine cell induces the effect. Okay, best known example of hormonal intracrine action is insulin, which acting via intracrine action regulates its own production and release by the negative feedback. Okay, so in case of high blood glucose concentration, insulin is produced and released. Most of the of insulin molecules are secreted by the beta cell to the surrounding extracellular fluid, but a small portion stays in the cell, bind to the intracellular receptor, causing the inhibition of insulin production and release. So intracrine action in most of the cases is used to regulate hormone production and release by the negative feedback. And finally, the last hormone action, mechanism of action, autocrine action. Again, there is only one cell, so the endocrine cell is the target cell. And in this case, the endocrine cell produces hormones, which are then released to the extracellular fluid, and they bind to the membrane-bound receptor. The hormone binding to the receptor stimulates the activity of the cell, mainly proliferation of the cell, which in some cases may lead to cancer development. This autocrine action is used mainly by growth factors. Okay, in case of endocrine mechanism of action, the hormones the hormones are required to be transported by blood to the site of the action. So, as I said, hormones can be divided into two big groups of hormones based on their chemical properties. So there are hydrophobic hormones and hydrophilic hormones. Let's start with the hydrophobic hormones. You know that the main compound of plasma is water. So the main compound of blood is actually water. That's why the way of hormone transport by blood depends on the chemical properties of the hormone. If the hormone is hydrophobic hormones, then the hormone cannot be transported freely by the blood. The hormonal transport requires blood proteins, albumins and globulins. Albumins, as you can see over here, are not specific. What is wrong? They are not specific. They are not specific proteins, so which means that they have medium affinity, or they show some kind of affinity to all hydrophobic hormones. So T3, T4, and steroids. Besides albumins, there are also globulins, and globulins are highly specific proteins. So they show high affinity to particular type of hydrophobic hormone. Because of that, we have several types of globulin, including CBG, corticoid binding globulin, which mainly binds cortisol, SHBG, sex hormone binding globulin, which binds testosterone and estradiol, so one of the estrogens, TBG, thyroid binding globulin, which transport T3, T4, and finally the last specific protein, vitamin D binding protein. As to hydrophilic hormones, they are water soluble, so they can be freely transported by blood. They don't require any blood proteins. Of course, there are some exceptions. There are three hormones, hydrophilic hormones, which are transported by blood proteins, like androgenetic hormone and oxytocin, mm, which are antidiuretic hormone and oxytocin, which are transported by neurophysics, and growth hormone, which is transported by growth hormone. Insulin-like growth factor binding protein. So it doesn't matter whether the hormone acts via endocrine, paracrine, autocrine, or intracrine action to induce the effect within the target cell. The hormone must bind to its proper receptor. So, um, so for now, the most important stage of hormone action is hormone binding to the receptor. We all know that there are several types of receptors uh, for um, for uh, for adrenaline and noradrenaline. I will look. I will quickly Google it. Wait. Um, okay. So as I said, there are several types of adrenergic receptors, as you can see over here. I will do it like this. Mm-hmm. Okay, as you can see, there is alpha 1, alpha 2, beta 1, beta 2 receptor, and although norepinephrine or epinephrine can bind to all of them, of course, with different affinity, which you can see over here. So the same chemical substance can bind to all of four receptors. The effects are different among they differ among the receptors. So, for example, alpha 1 receptor leads to vasoconstriction, whereas beta or 2 receptors causes vasodilatation. Mm, so, in general, we may say that the most important stage of hormone action is hormone binding to the receptor. And as I have already told you, there are two main types of hormone receptors: membrane-bound receptors and intracellular receptors. Membrane-bound receptors are the receptors for hydrophilic hormones, and intracellular receptors are dedicated for hydrophobic hormones, including T3 and T4. Let's start with the membrane-bound receptors. In the class of membrane-bound receptors, we have receptors which are linked to G protein, so protein G linked receptors, which we talked about a lot during cardiovascular system physiology tutorials. And there are also enzyme-linked receptors. So let's start with the G protein coupled receptors. As you know, there are several types of G proteins, including GI protein, GS protein, and GQ proteins. After hormone binding to G protein coupled receptor, the beginning of the mechanism, of action is the same for all G protein coupled receptors. So, as you know, in inactive states, the receptor is bound to G protein, and alpha protein is connected to GDP molecule. After hormone binding to the receptor, the GD molecule is replaced by GTP, and because of that, the alpha GTP subunit dissociates from beta and gamma complex and migrates, travel, and is released to the cytosol. Beta and gamma subunits, on the other hand, they stay connected with to the cell membrane. So the alpha GTP subunit of different G proteins triggers different cellular response. So as you know, the alpha GTP subunit of GS protein, it activates adenylate cyclase, and because of that, the concentration of cyclic AMP, intracellular concentration of cyclic AMP, increases, which activates protein kinase A. The phosphorylation of different proteins by protein kinase A triggers the effect within the target cell. The second GI protein, acts somehow opposite to GS protein. I stands for inhibition. The alpha GTP subunit of GI protein inhibits the activity of adenylate cyclase. And you know that because this enzyme is no longer activated, is no longer active, the rate of cyclic AMP production decreases. Moreover, besides adenylate cyclase, there is also another enzyme which is activated, but this time by beta and gamma subunits. Beta and gamma subunits, they activate phosphodiesterase, which degrades cyclic AMP into 5' AMP, 5' AMP, which further decreases the concentration of cyclic AMP, and therefore decreases the activity of protein kinase A. Okay, and finally, GQ bound receptor, the alpha subunit of GQ protein, alpha GTP subunit, it activates phospholipase C beta, which you can see over here. So alpha subunit activates phospholipase C beta, which converts PIP2, so phosphatidylinositol 4,5 into 2-second messengers IP3, so inositol 1,4,5 3-phosphate. You may say inositol 3-phosphate and diacylglycerol. IP3 is released to the cytosol and is a, a very strong agonist of ryanodine receptors. Okay, of ryanodine receptors. IP3 binds to ryanodine receptors, opening the channel protein and therefore causing calcium influx into the cell. Okay, and now calcium ions that are released from sarcoplasmatic reticulum, then they bind to calmodulin, and together with calmodulin, they activate calmodulin dependent protein kinase. And you know that in some cases, the activation of calmodulin dependent protein kinase A leads to a phosphorylation of myosin chains and therefore is responsible for the vasoconstriction, like in case of angiotensin II. Okay. Besides binding to calmodulin, the calcium ions that are released from sarcoplasmatic reticulum, they act together with the diacylglycerol and they activate protein kinase C, which can phosphorylate membrane proteins or intracellular proteins, inducing the effect within the target cell. And so over here, you have the list of hormones and their respective receptors. I see over here that I am lacking information about TSH. So TSH receptor, actually there are two types of receptor for TSH, GS bound receptors and GQ bound receptors, and TSH must bind to these two receptors to induce the effect. So to induce production and release of T3 and T4. And angiotensin receptor, AT1 receptor, is bound to. So as you can see, the list is not complete. So there are so many receptors that it is difficult to put them all in the list. But over here you have the most common receptors and hormones. Okay, but does mean that G, because we talk a lot about the G bound receptor, does that mean that is it possible that G protein bound receptors are not only, is it possible that they are not only used by hormones, but also by other chemical substances like toxins? It is. So there are at least 3 toxins that interact with G protein bound receptors, inducing the effect. So, as you can see over here, the cholera toxin and enterotoxin of Escherichia coli, they bind to GS bound receptor, increasing the activity of adenylate cyclase and therefore increasing the activity of cyclic AMP. Whereas Petricius toxin, it acts via GA bound receptor, inhibiting the adenylate cyclase activity, increasing the concentration of cyclic AMP. The second group of membrane-bound receptors, the second group is enzyme-linked receptors. And again, there are two subtypes of enzyme link receptor. So the enzymes which have intracellular enzymatic activity and receptors which are associated with the enzyme. The best example of an enzyme of the receptor which has intrinsic enzymatic activity is insulin receptor. So as you can see, the insulin receptor it has two different domains: extracellular domain, transmembrane domain, and intracellular domain. The intracellular domain of insulin receptor is in fact an enzyme, tyrosine kinase. So after insulin molecules binding to the receptor, first the receptor translocate within the cell membrane and form dimers. After dimerization, the insulin receptors undergo autophosphorylation, and this phosphorylated proteins, phosphorylated tyrosine molecules within the intrinsic domain of insulin receptor, they act as the binding site for insulin receptor substrate molecule IRS1 or IRS2, and insulin receptor substrate molecules, they are proteins that after binding to phosphorylated insulin receptor, they exert biological activity which activates different proteins, mainly different protein kinases. And because of that, a whole chain of protein kinases is activated, and the phosphorylation of proteins within the target cell induces the effect of insulin. Like, for example, increased uptake of glucose, or even increased production of membrane proteins which are responsible for glucose intake, like GLUT transporters. As I said, the second subgroup of enzyme-linked receptors are the receptors that are actually connected with actual enzyme. Like in this case, This is the receptor for atrial natriuretic hormone. As you can see on this graph, the receptor is bound to guanylate cyclase, which converts GTP into cyclic GMP, uh, a second messenger, which is used by which maybe not used, but which causes increased sodium excretion by kidneys, and because of that, it increases the urine formation and urine volume. And finally, the other example of the hormone which is linked to enzymatic receptor is a growth hormone receptor. Growth hormone receptor is a very particular type of receptor because although this is a membrane receptor, the growth hormone binding to the receptors directly influences the gene transcription. So as you can see, the growth hormone molecule has two binding sites for the growth hormone receptor. So two receptors are bound by one growth hormone molecule. The hormone binding to the receptors leads to the activation of Janus kinase activity, and because of that, the receptors undergo autophosphorylation. The phosphate within the phosphate group, which is now connected to the receptor, they activate different enzymes like STAT kinases, or even are bound by insulin receptor substrates or co-activator activator, and this results in insulin-like growth factor 1 and 2 production and release in liver, regulation of glucose metabolism, for example, by increasing the number of insulin receptors, by regulating insulin receptors in the target cell, or inducing transcription and translation of different genes. And finally, let's talk about nuclear receptors. There are two subtypes of nuclear receptors: steroid hormone receptors and thyroid hormone receptors. Let's start with the steroid hormone receptors. The receptors for steroid hormones are located within the cytosol, as you can see over here, and they are bound with HSP protein, so heat shock proteins. Heat shock proteins. We may say that heat shock proteins are protection which doesn't allow receptor translocation to the nucleus without deactivation or without binding the steroid hormones. So, sorry. Oh my goodness. Okay, so again, cell membrane, nucleus membrane DNA. So as I said, the steroid hormone receptor is located within the cytosol, and it is bound with heat shock proteins. Okay, so first the hormone reaches target cell, and because of the hydrophobic properties, the hormone can freely pass through the cell membrane and then bind to the receptor. Because of the hormone binding to the receptor, the heat shock proteins dissociate from hormone receptor complex, and now the hormone receptor complex can pass through the nucleus membrane and then bind to a specific fragment of DNA regulating gene expression. So we may say that the hormone receptor complex is in fact a transcription factor. Okay. Recent studies have shown that steroid effect can be observed very quickly after steroid administration. So, for example, cortisol or its derivative, hydrocortisol, can induce effect within the target cells in 30 seconds. This means that intracellular receptors are not that besides the intracellular receptors, there is also other type of steroid receptors. So right now I would like you to look at this graph, not this one, this one. Okay, so as you can see over here, let's focus on this part. Okay, over here you may observe the effect of not the effect, but the mechanism by which glucocorticosteroids affect bronchial epithelial cells and vascular smooth muscle cells. Let's start with the bronchial epithelial cells. So the glucocorticosteroids, they bind to GS bound receptor, increasing the activity of adenylase cyclase, increasing the activity of protein kinase A. Dephosphorylation of SERCA pump reduces the concentration of calcium ions inside the cell, and that's why glucocorticosteroids binding to a GS bound receptor leads to different response of bronchial epithelial cells compared to, for example, to vascular smooth muscle cells. In case of vascular smooth muscle cells, glucocorticosteroids bind to GI bound receptor. Sorry, GQ bound receptor. Because of that, we observe increased concentration of IP3, which leads to renin receptor channel protein opening, increasing intracellular calcium concentration. Because of increased calcium ions concentration inside the cell, we observe vasoconstriction of the smooth muscles. So as you can see, glucocorticosteroids can not only regulate gene transcription, gene expression, but they also can bind to membrane-bound receptor, inducing non-genomic effect. However, the non-genomic effect of glucocorticosteroids, non-genomic effect of steroid hormones is still under in the process of confirming. So we know that there is non-genomic action of steroids. However, we do not fully understand the importance of this action. And finally, thyroid hormones receptor. And before we go through this mechanism of action, please remember that the thyroid hormone receptor shows high affinity to T3. So basically, only T3 can bind to nuclear T3 nuclear thyroid hormone receptor. We assume that thyroid hormones T3 and T4 are hydrophobic, so they can freely pass to the cytosol through the cell membrane. T3 is deiodinated, so T4 is converted by deiodinase the second to T3, and then T3 passes through nuclear membrane and binds to the receptor complex consisting of retinol acid receptor and thyroid hormone receptor. After T3 binding to this receptor complex, co-suppressors proteins dissociate from the receptor, and co-activators bind to the complex of the receptors. Co-activators binding to thyroid hormone receptors, retinol acid receptors, it leads to chromatin acetylation, which relaxes the structure of chromatin within the histones and therefore promotes gene transcription. For example, because of thyroid hormones binding to nuclear receptor, there is more sodium-potassium bound within all cells, target cells, more sodium-calcium exchanges. Moreover, T3 increases the number of funnel channels within the pacemaker cells in the cell. So T3 binding to the complex of thyroid hormone receptor and retinol acid receptor promotes gene expression. But of course, besides the genomic mechanism of action, T3 also exerts T3 and T4 also exerts no genomic action, which you can see over here is somehow connected to integrin receptors. And now we know that T3 T4 binding to different membrane-bound receptors activates different protein kinases, like this, and phospholipases, inducing the effect, which is also connected with regulation of gene expression. However, it is induced through membrane-bound receptor. Okay, so I think that we discuss all the most important receptors. Next week